at least for one of the compounds I'm going to show you. There were lots of questions that came from you about what we have learned so far from the trial. So let me just say that for those of you who asked about the Riluzole trial, we're just not done yet, so I can't show you. The Toronto site has finished, but the Hamilton site and the London site are still recruiting until the end of June. So hopefully by mid-fall, we'll be cleaning up the data and sharing with you the results. There were some questions about the pioglitazone study. Also, it hasn't finished. But I will show you the results of the omega-3 study, and I will show you a little bit of early data on oxytocin, but not the final data. All right, so I put the autism slide up, but I want to make a point that's not an autism point. So yes, if you put that slide up, social communications and repetitive behaviors make up autism, but the types of symptoms and the types of difficulties kids have when they come to clinic and request medication almost never are about social communications and repetitive behavior. Sometimes they are, but almost never are when they actually request medication. So kids come in because they have ADHD-like symptoms and because they have aggression and anxiety and emotional dysregulation. They come in because they have upset stomachs and they're constipated. They come in because they can't sleep. They come in because they have immune differences, right? So we can classify our disorders any way we like. At the end of the day, all of these symptoms present, various combinations of these symptoms present in all our kids, whether their primary diagnosis happen to be ASD or ADHD or OCD or whatever have you. Looking back to the overlapping biologies and phenotypes. And the current approaches for developing medications, as we've discussed before, have not been translational. And so by that we mean we didn't figure out what is the molecular biological difference in this kid's brain and then develop a medication or another treatment to go after it. We've done it a little bit in ADHD, and that's why the stimulants are so successful, but it's not, that particular deficit is not the only deficit in ADHD, as Russell pointed out, and lots of kids actually don't respond. We have a little bit of that in OCD with the SSRI, so some of the repetitive behaviors of OCD seem to be primarily related to serotonin dysfunction, but Paul will tell you that's not true either, that there is glutamate and GABA and other contributions to OCD. And ASD has been the worst of the bunch. We don't have a single medication that has been approved for the treatment of core symptom domains because we've never translated from an autism molecular deficit to treatment. Now, we're just starting to do that, and PONT is part of a series of efforts internationally, for those of you who asked before about international efforts, to actually start translating from the basic science down to treatments, and also to be looking across domains as we're doing here with the variety of disorders included. So in order to develop effective treatments, we need to understand the answer to two questions. What is the biological difference? What is it that is different about these brains, about the chemistry of these brains, about the structure of these brains, or the bodies, because we talked about the gut and the immune connection. And we need to understand where the difference is so that we can actually develop treatments that go after that particular difference if we choose to treat it, and that's a whole different discussion, and to do it in as targeted way as we can. On the genetic side of things, and Nii presented to you this morning, the truth is we're like hundreds and hundreds of genes now have been implicated across these neurodevelopmental disorders. All of them are, many of them are overlapping, but it becomes a bit of a daunting task, right? So if you have, let's say in the case of autism, 450 so far, candidate genes, how do you develop drugs for 450 different differences, right? But it turns out that it may be a little bit easier, as we've talked before. So it may be that not all of these biological differences are completely distinct. They may be mapping down to pathways, and they may be molecular pathways or brain pathways, like Dr. Taylor showed you. And it may be that once we figure out what their pathways are, we can target them better. And there is a little bit of effort to do that. So this is a synapse that people showed you before. You have your first neuron, your second neuron, and these are the elements of the postsynaptic space, the space that's after the neurotransmitter has done its job and the signal has gone down to the second neuron. And what I want to show you is that there are plenty of targets in that postsynaptic space that are actually relevant across these neurodevelopmental disorders. And we may have many gene mutations that map onto each one of those targets. So if we can develop treatments for those targets, not the original genetic mutation, we may be able to treat a multitude of genetic mutations with the same treatment, but thoughtfully, right? We know what the pathway is, we're not guessing anymore. So I can highlight how this is absolutely not autism specific for those of you who are not autism families in the audience. So on the far my right, your left, you see the glutamate manipulation. So Paul presented a couple of minutes ago and told you about riluzole and memantine being trials in OCD, and I'm presenting and I'm showing you riluzole and memantine being trials in ASD. So these targets again are overlapping. We learn from one disorder, really transferable to the other disorder. What I'm going to show you today is not going to be on this model, but I want to remind you of this model because some of you are going to get invitation letters in the summer for a new trial of the Anagnostou compound, which is on this side of the screen, which is also affecting part of this pathway in the postsynaptic state. So if you get if you get a letter that says something about Anagnostou, this is what it is about. But I'm going to focus on something else. So one approach is to take the synapse and targets that are coming from genetics. Another approach is to look at other targets. Another target would be inflammation. And so this is a slice through the cerebellum of a child who passed away of other causes. And if my thing was working, I would show you that there is quite a bit of inflammation. So let's say on the picture that's closer to me on the bottom panel, you have a little vessel and you have infiltration of inflammatory cells all around it. In the picture above that, you see these big, plumpy, pink cells. They're called the Purkinje cells in the cerebellum, and there's just not a lot of them. They've been eaten up with a lot of inflammation around. We know that at least in a subgroup of kids with ASD, inflammatory processes are important, and so is the case probably across neurodevelopmental disorders. And the light gray is the placebo, and the dark gray, blackish is the drug. So if you look at panel A, the first line was our primary outcome measure, which is the composite autism score for the PDDBI, so their overall autism severity. These are toddlers between the ages of 2 and 5. You will see that actually placebo did better than the drug. So there was a bigger decrease in the autistic-like behaviors on placebo than there was on the drug. And the difference, though, was not statistically significant, so we call that an equal response. If you go down to B, this is the BASC, and it measures internalizing and externalizing behaviors in addition to other things. Externalizing behaviors, which is the last set of lines in that block, was our second primary outcome measure. Externalizing behaviors include things like irritability, agitation, hyperactivity. You will see again that the placebo group actually improved over the study, and in this case, the omega-3 fatty acid group actually worsened over the study. This is why it's very important to do double-blind placebo-controlled trials. I can tell you that when we used a global improvement score, when we and the family discussed the case without knowing what the child was on and decided to assign a score that would say whether they were a responder at the end of the study or not, 37 of the 38 kids were rated as responders. So we all thought we were doing amazing in this study until the data came out, right? So of course kids did better. Over 6 months, kids improved. These are young children. They're naturally improving. Now, adverse events were not significantly different, so there was a little bit more upset stomach, as you would expect with the omega-3 fatty acids in the kids who were exposed to the omega versus the placebo, but it was only a trend. But then remember the discussion we had before about figuring out stratification. So if omega-3 fatty acids work at decreasing inflammation, then you may expect kids who have high inflammatory markers to be more likely to respond than kids who don't have high inflammatory markers. And so we looked at that in an exploratory way, and we found no evidence that their baseline inflammatory status would predict whether they would be a responder or not. So at this point, although this was a small study, the direction of the effect is the wrong direction. So we will not invest further in omega-3 fatty acid studies. Now, this is not about nutritional supplementation, right? So there's plenty of data to say our diets are low in omega-3 fatty acids. We supplement infant formula with omega-3 fatty acids. If your child doesn't eat fish, there are recommendations for providing omega-3s, but that's 1/10 of the dose of this. So our dose is treatment. It's what autistic kids typically take from a variety of companies as treatment for autism. This is not just your general nutritional supplementation. Nobody would speak against that. Ideally, your children get it from food because we're not quite sure the supplements work the same way, but if they don't, they should get their appropriate 30 to 50 milligrams of omega-3s, which is the current recommendation for nutritional supplementation. But we have no evidence to suggest that you should be investing in high-dose supplementation with omega-3 fatty acids at this point, especially if your child is a toddler, because for that particular group, we're getting a little bit of a worsening effect. Now, there is one more caveat I want you to know. Because the sample was small and I was disturbed by the finding, I wanted to make sure that there was nothing special about the kids who were in the active group, in the drug group, that made them more likely to have a bad outcome. So we looked at every variable we could find to see if there is anything that was different between the two groups that may not have been randomized properly, that may have predicted the outcome. And the only thing we found is that the kids on the active group may have had more gastrointestinal complaints at baseline. So this is a little caveat. So it may be that the kids who are getting worse are the kids who are having the gastrointestinal complaints to begin with. Our study is not designed to study that, so this is just a caution that we did see this difference between the two groups. So very quickly now, I got lots of questions for you about the outcome of the oxytocin studies. How many people have never seen me talk oxytocin before? Everybody? A couple of people. Very quickly, oxytocin is a hormone. We produce it in our brain. All of us have it. We thought it was a girly hormone. We thought it was only appropriate for women because it helps with delivery and milk letdown and breastfeeding. Turns out men have plenty of oxytocin in their brains. We were trying to figure out what oxytocin does in people's brains in the animal studies. And this is another example of how mice studies can be extremely important in our understanding. People noticed that that's too bad. People notice that when you introduce a new mouse to a litter, if they have an intact oxytocin system, they will sniff the other mouse for a certain amount of time. And then the next time in the orange line that you reintroduce the mouse in the litter, they will keep sniffing less and less because now they recognize that mouse. In the case when you knock out the oxytocin receptor, every time you introduce the mouse in the litter, they spend the same time suggesting that either they don't recognize that they know this mouse or that that doesn't translate in change in social behavior. So there are tons of studies like this with variable paradigms to suggest that oxytocin is involved in aspects of social perception and social cognition and affiliation, attachment across species in the animal kingdom. There were some early studies to suggest, in college students, to suggest that even if you don't have a deficit, if you take an oxytocin puff versus a placebo puff and you look at stimuli like this, and you have to guess the emotion involved, that you're better at detecting emotions from stimuli like that if you're exposed to oxytocin than if you're exposed to placebo. So based on that, a series of very early human trials started. So those of you who are in the room who participated have either participated in the open label study or in the randomized study. The open label study has been finished and it got published this year. And just to remind you, what the open label study did, according to the request of Health Canada, is to test whether there is a maximum tolerated dose to see if there is toxicity to the compound. So kids were exposed for 3 months to increasing dose of oxytocin, and they were evaluated over 3 months, and then they were taken off the drug and brought back 3 months later and were re-evaluated again to look at the efficacy and the safety of the compound. This data we have. So I want you to know there is no control group here. Remember what I told you about the omega-3 study? We all thought they were all doing great, but they were not all doing as good as expected. So when we do open label studies, we have to be very, very, very careful not to overinterpret the results. But if you see p-values in the 0 to 12 that are less than 0.05, we considered it as a trend suggesting that it was worth continuing with a randomized study. So what you see on the left column is a series of measures. You don't even need to know what they are. Those of you who are part of the study had to do them. But they are questionnaires for the parents about the kids' social function. They're computer games for the kids themselves to do, to actually measure their ability to detect faces and emotions, match faces, match emotions, those kinds of things. There was some irony and empathy tasks where kids were told stories in a voice that didn't match the content, and they had to figure out whether the person meant what they said or whether they're being ironic. And there were some measures of anxiety, and we saw nice improvements across domains with oxytocin at 12 months. And then we stopped the drug and brought them back, and we saw that some of those improvements were maintained. Not the anxiety ones, but the social gains were maintained. There were some side effects. Most of them, again, we have no placebo, so we can't compare yet to see if it's more frequent in oxytocin. But the ones that we're a bit worried about, well, not really worried, but you should know about, is that there's probably an appetite effect. So there were enough kids who had a suppression of appetite that we need to follow that up. There were some effects on mood. So although the whole group got less irritable with time, there were a few kids who got more irritable. There is a biological plausible explanation for that, so we have to be careful. The randomized phase now was the phase that was supposed to be done as a follow-up, where kids get drug or placebo. We just finished enrollment, so all the kids who are supposed to be in the study are in the study. We're going to finish by the end of August, so we should have the final data for you by mid-fall. The other thing to know about is the Canadian Institute of Health Research got interested in this concept, and so we just started an adult study of oxytocin. And so between us and McMaster, St. Joe's, so it's going to run for the next 2 years, and we need 140 adults who are relatively high functioning to come to the study. They will be randomized to drug or placebo to see if the stuff actually is as good as it looks in the open label studies. So I'm going to stop. This is the lab at Bloorview, but many labs are actually involved, and the funding comes from many people, and we are grateful to our funding sources, but we're mostly grateful to you, obviously, because without your participation, we cannot answer any of those questions. So maybe I'll take a couple of quick questions, and then I'm going to do some final close-up housekeeping things.